So that's the last of my bad French. I do apologize. So, but I can understand French, but speaking it, I'm terrible. So I'm going to start off with, I'm going to set my clock to make sure I don't talk for more than half an hour. I'm going to start off with the most important slide, and that is the acknowledgements. So this work was recently published. Well, I'm going to present some published work just to bring those of you up to speed who haven't read it, and then I'm going to publish some unpublished work. So Natalia is the PhD student who published the Nature Genetics paper. She is writing up her thesis at the moment, and Lily Wu was the second author on that paper. It's her unpublished work that I'm going to be presenting later. None of this would be possible without an absolute amazing postdoc, Joe Chambers, in the lab. He's more than a postdoc. He's basically another PI in the group. And talking of PIs, this is a collaboration between 3 departments and 3 labs. My lab is the antitrypsin lab, working on the cell biology. Peter Campbell is an expert in sequencing, and Matt Hoare, some of you may know, he's an academic hepatologist in the UK. He's particularly interested in changes in early cancer. I don't need to introduce this particularly to this audience because we've also had fantastic talks today about it, but just to justify why a respiratory a pneumologue is speaking at a liver audience, this is a disease where we both, the respiratory doctors and the liver doctors, will see these alpha-1 antitrypsin patients in our clinic. So alpha-1 antitrypsin is a very abundant protein. You probably make about 5 grams a day and secrete it into your blood. It circulates, and it's a serine protease inhibitor. Do we have a pointer? Or can I get the pointer to work? Yeah, so this is the beautiful protein. It's called alpha-1 antitrypsin. It works as a protease inhibitor because it masquerades as a substrate. This loop here, I'm afraid some of these structural features you need to know for the rest of the talk. This loop here is called the reactive center loop, and it looks to a protease like a substrate. The substrate cleaves it, but and it forms a transient covalent bond. But normally that will be released, and the protease will go off and cleave something else. This is a metastable structure. So when the protease, oh dear. When the protease cleaves there, that strand inserts into the underlying beta sheet. This is a very stable structure. It's more stable than the secreted antitrypsin. And as a result, it releases energy, which is used to denature the protease. And that's how the enzymes work. However, there are mutations that alter the folding of alpha-1 antitrypsin. The most common we've heard about today, the Z mutation, is in this portion of the protein, and it interferes with the folding within the endoplasmic reticulum. Not so much that it activates endoplasmic reticulum stress, but rather it leads to a very strange folding pathway. We know an awful lot about the folding pathway of this protein. As it enters the endoplasmic reticulum, so it's made by ribosomes in the cytosol, it passes through a pore in the ER membrane, and within the endoplasmic reticulum, it has to fold. The final folding step is the insertion of this C-terminus into the second beta sheet here. That's necessary to nail down this reactor center loop. So this is the critical final folding step, but it's delayed in patients who have the Z mutation and a number of other rarer mutations. Because that pocket that the C-terminus should insert into is open, it can receive the C-terminus of a neighboring molecule. So you can see we've now formed a dimer within the endoplasmic reticulum. To the cell, this looks like a relatively well-folded protein, but it's an incorrect dimer. But critically, when this happens, the reactive center loop inserts into the underlying beta sheet, generating that hyperstable conformation, which the cell can't unfold. But more importantly, it then positions the C-terminus, which is here, to insert into another molecule. So you can see how we generate long polymers that look to the cell like a properly folded protein, but are in fact completely inert. They can be degraded, but over time in your patients in the liver clinic, you'll see them accumulating. And if you do a liver biopsy, and we've seen many of those today, that you see the inclusion of these periodic acid shift positive globules. They are actually the endoplasmic reticulum filled up with these polymers, which are inert. So by themselves, they're not causing stress, but they do make the cell more sensitive to other forms of stress. We don't fully understand the biology of this disease, even though people have been studying it for about 40 years now. The first electron micrographs of these polymers dates back to the early 1990s, but we still, until recently, we didn't even understand the polymerization mechanism. So we approach this, and the work that we've recently published is trying, can we use naturally occurring somatic mutations that are happening in our patients to tell us something new about the biology of this disease? We normally think of somatic mutations as being bad. We all know that cancer is caused by somatic mutations, but they're not always bad. And there's a lovely speculative paper by two French scientists in the 1970s who published a paper where they were speculating that if a cell is in a regenerative tissue, so they were thinking more about the blood and bone marrow, but the liver is particularly regenerative tissue, and you apply a selection pressure to it, there is nothing to stop these cells accumulating somatic mutations that over the life of the organism make these cells more fit. And we know that somatic mutations can be beneficial because if we didn't have somatic mutations, we wouldn't have an immune system. That's more directed somatic mutations. We're talking about more stochastic mutations. So Peter Campbell's group for many years has been looking at the liver as a place where somatic mutations occur and hepatocytes can accumulate somatic mutations that make them more fit. This is not driving them towards cancer, it's making them survive toxic environments. So for example, these mutations, if they occur in a particular pathway or in a particular gene, this is biologically useful for us because we can identify the pathways that are important to the hepatocyte to survive in those conditions. So for example, in metabolic stress, hepatocytes accumulate somatic mutations that reprogram the signaling and metabolic pathways to make the hepatocytes survive metabolic stress more. And this is not pro-cancerogenic, this is pro-hepatocyte survival. It's also been described in liver diseases where there are germline mutations which are toxic to the cell. So for example, this is a ribosome assembly factor, but imagine you had a somatic germline mutation which is toxic to the cell, but the organism is able to live with this damage. If we acquire a somatic mutation in the same gene that suppresses the toxicity, it will tell us something about how to suppress that toxicity. Or if we identify somatic mutations accumulating in downstream targets, it tells us which pathways we have to manipulate in order to help the hepatocyte survive this germline mutation. And this has been done for a ribosome assembly factor disorder in the liver. So we asked this question: are there somatic mutations accumulating in the livers of our patients with alpha-1 antitrypsin deficiency? Because the liver disease is unusual. All of my patients with alpha-1 antitrypsin deficiency are going to develop emphysema. If they're sensible and they don't smoke, they might get end-stage lung disease when they retire. So these are non-smokers. If they do smoke, they can require transplantation by the time they're 40. So that's a very, very penetrant phenotype. That's the loss of function phenotype. Interestingly, the toxic gain of function phenotype within the liver is less penetrant. We've heard already today that only about 25% of these patients will develop clinically relevant liver disease. Liver failure only accounts for 13% of deaths in this condition. We also know, and there's some wonderful, I'd recommend you to read some of Marion's papers, we know that there's a big difference between the liver disease that accumulates in children and the disease that accumulates in adults. So why is it children survive this condition, and how is it so many? How is it the majority of your patients, well, my patients with Z antitrypsin deficiency don't become your patients with liver failure? So to address this, we did a simple experiment, very expensive, but simple experiment, is we took 10 livers. 5 of them were explants from patients who had cirrhosis due to hemochromatosis, and that was our control group. We wanted a simple cause of cirrhosis, and we also had 5 livers of patients who had end-stage liver disease because they were homozygous for the Z allele. And this is Natalia's work. Natalia took these livers, took samples from across the liver, made blocks, sectioned them, and then laser microdissected multiple little biopsies within each sample. So we have dozens of biopsies from across each of these livers. We then sent each one of those microscopic biopsies for either whole genome sequencing or whole exome sequencing being a bit cheaper. And this allowed Natalia to catalog all the somatic mutations in these 10 patients' livers. So this was a huge amount of work and an awful lot of sequencing. But I suppose the £100,000 question is, do we see somatic mutations in either of these diseases that are occurring more often than by chance? And with hemochromatosis, the answer is no. There's nothing. Nothing that stood out, at least with this number of patients. But this was the first of several jaw-dropping moments with this project for me, and that is when we looked at the patients with alpha-1 antitrypsin, they all, many, many times in different places in the liver, evolved mutations within SERPINA1, which encodes alpha-1 antitrypsin. The p-value being a sort of crazy 10 to the minus 20. This is happening many, many times in every single patient. So just to illustrate this is one of the little bits of liver that Natalia took from one of the explants. So it's a microscopic slide. Each one of the dots is one of the little tiny punch biopsies she laser microdissected. And what we've done, and then they're all sequenced, and then they're color-coded by sharing the same somatic mutation. So if we look at where as you're looking at it, the far right side, this little orange patch contains 4 biopsies, all of which share the same mutation, which is a nonsense mutation. It's the acquisition of a stop codon at this particular position. Okay, and every other colored patch on that tiny area of liver has a clone of cells which has accumulated a different SERPINA1 mutation. We don't see this for any other gene in this disorder. So this is this is happening far more likely than chance. So it's telling us something about the biology. Now you might think, okay, well, all it's telling you is if you destroy this gene, you protect the patient from the cell. If that were the case, that would have been slightly disappointing. So the second jaw-dropping moment of this project is where we looked at where the mutations are. And whilst we do see some mutations in the 5' half of the transcript, almost all of the mutations are in the final exon. This is that C-terminus that I was so excited about. This contains the little part that inserts at the very end of folding. It also includes the reactive center loop, that little part of the protein that the alpha-1 antitrypsin uses as a bait to catch. So it seems to be, this genetics is telling us, it seems to be beneficial to mutate this final exon. All of these mutations are truncating. So everything that's in green is, I'm sorry about the 6% of men in the audience who are colorblind, but all the ones that are in green are frame shifts, and all the 2 that are in red are nonsense mutations. They've got stop codons. These are the ones that Lilly chose to study because they're the simplest. They represent the truncation, but we don't have to worry about acquisition of extra amino acids. So these are the things that she decided to study. I'm very familiar with this protein. I live it every day, but just to orientate you again, what we're talking about is this is the C-terminus, which is necessary for the folding. It normally inserts into this beta sheet. In the polymerization process, there is a domain swap, so the same C-terminus inserts into a neighbor. The 2 mutations that we're seeing, either the long mutation just removes this final C-terminus. You'll notice that it still has the reactive center loop that can insert into the beta sheet and generate a hyperstable conformation. The other mutation, which is up here, We'll also delete the C-terminus, so we can't have the domain swap, but now we can't have that hyperstable insertion of the 6th strand. So taking these two representative mutations, a longer one and a short one, but all delete the C-terminus, we asked if they have any functional effects on the cell. So one of the things that we do in the lab is we express these proteins in cells and see what they do. It's well known that if you express wild type M antitrypsin, and it's expressed here in magenta, it doesn't express, you can't see it very well with the lights on, but what you will be able to see is that most of the intracellular protein is in this location in the cell, this little lump next to the nucleus, and this is the Golgi apparatus. This tells you that it's trafficking out of the endoplasmic reticulum, it's gone to the Golgi, and it's going to be secreted. By contrast, the Z protein, the normal toxic protein, doesn't get to the Golgi. There's no Golgi apparatus to be seen here, and those with very good eyesight will see that the endoplasmic reticulum has this very same strange structure. It's broken up into dozens of little globules, very much like the liver biopsies of the patients you see. So what we can count these cells. If you express any ER protein, you can cause some vesiculation. So it doesn't go from 0 to some, it goes from some to lots. So we normally have a difference between M and Z when we express these in cells. What happens if we do the C-terminal truncation? Although we do see a few inclusions, most of the endoplasmic reticulum is rescued to normality. And if we count this, if we can't count large numbers of cells, we find that this C-terminal truncation turns the Z. So they've got the Z mutation, but we've then taken off the C-terminus. We've now turned it into a protein that doesn't cause these vesiculations. So what's happening to the protein? It could either be being degraded or secreted. So if we look at the medium that these cells are growing in and we measure the antitrypsin that they're secreting, not surprisingly, the M secrete lots of protein, the Z secrete very little protein, so that phenocopies what we're seeing in our patients. But when we have a Z and we delete the C-terminus, we completely abolish secretion. So we're not, by removing the C-terminus, making a protein that the cell can secrete, but we're also making a protein that doesn't cause damage to the endoplasmic reticulum. So what's happening? If we take the cells now, this is what's inside the cells now, and we run them on a denaturing gel, we can see that if we separate the lysates into the soluble protein and the insoluble protein. So you simply take the lysates, centrifuge it, take the very large insoluble aggregates and call that the insoluble fraction. We see M inside the cell, but we know that's going to be trafficked. We see some Z in the soluble fraction, but a lot of it enters this insoluble fraction as we know it aggregates within the cell. We also see some of our truncated proteins, less in the cytosol, and we see none in the insoluble fraction. So we're preventing it from undergoing a phase transition to a solid. We can take the same samples and run them on a native gel, which allows us to look at not simply just the size of the protein, but the conformation. And you'll see if we block with an antibody to total antitrypsin, M forms a nice monomeric band. This is protein that's well folded and is going to be secreted. The Z forms this ladder of polymers, and we see that the material in the insoluble fraction is all polymers. We can use an antibody which was generated against polymers and recognizes the polymers inside patients. And we see that our Z causes polymers both in the soluble and the insoluble fraction, and that's completely abolished in our mutations. So it's not accumulating, and what's happening to it? It must be being degraded because it's not being secreted. This experiment, Lily takes the antitrypsin protein. Now, when I was little and I was a postdoc or a PhD student, you would radio-label the protein, wash away the radio-label, and see how long the radioactivity lasts in the cell, called a pulse chase experiment. Students don't like doing that anymore. They don't want to get irradiated. So Lily came up with this rather cool approach, is she fused antitrypsin to a protein called HALO. This is a bacterial hydrolase that we often use as a tag. It binds to haloalkanes. So say if you've got a polyalkane with a chloride on the end of it, it will covalently link to it. It's very useful in the lab because we can fuse it to various fluorescent molecules and use it as a nice way of fluorescently tagging all of our proteins. What Lily's done, though, is she's added her fluorescent protein, washed it away, and then sees how long the fluorescent protein lasts in the cell. So it's a non-radioactive pulse chase. And actually, you don't even need to do a Western blot. You just run the gel and slam the gel on the detector, and the protein fluoresces. It's lovely. So we can see that both of these truncated proteins are degraded much, much faster. Z lasts in the cells for more than 24 hours. These Z with a truncation are degraded in minutes, actually. This allows us to do various experiments where we can look at the mechanism of degradation, and she can inhibit different degradatory pathways. So if we inhibit the lysosome with a drug called baflomycin, it doesn't do anything. If we inhibit the proteasome, we stabilize it. So partway through, sort of 2/3 of the way through, we've got this simple model. Normally, M is secreted, Z accumulates as polymers, but Z, when we delete the C-terminus, just gets degraded by the proteasome. Lovely, nice story, published. But it can't be the whole story. Because if we look at those, we go back to Natalia's sequencing data, we see that the variant allele frequency is always less than 50%. We never see homozygosity. In these clones, we always see that allele frequency makes up no more than half of the protein in the cell. So that means we must have a copy of Z and a copy of Z with one of those truncations. And in order to model this is unpublished data, what Lily has done is she's made bisostronic vectors. So these are plasmids which can make two proteins, one of which will be a fluorescently tagged Z antitrypsin. The second one will be whatever she wants it to be, either another copy of Z, another copy of M, or one of these truncations. And this is where it becomes, to a cell biologist, this is fun. I mean, it's difficult to express how fun this is, because usually when every gel you show, you lose half the audience. So I'll try and make this sound exciting. When you express Z with another copy of Z, it's retained in the cells, just like it's seen in our patients. If you express Z and another copy of M, you get fewer polymers, but they still last a long time. And so we know that MZ patients are overrepresented in your clinics. But if you take Z and then add one of these truncations, the Z gets removed from the cell very quickly. So it's having a dominant effect. So it isn't simply we're removing a Z allele, we're adding something that is allowing the hepatocyte to degrade this material. So that's the whole cell. If we look at the insoluble fraction, it's even more dramatic. So again, if you look, lots of the Z goes into the insoluble fraction. In a cell that's expressing M and Z, less of the Z goes into the insoluble fraction, but some does. If this material enters the insoluble fraction, it stays there for 24 hours or more. But again, when she includes one of these truncating mutations, it's allowing the cell to degrade this material. We can do the same experiment where we look at the inclusions, and if we have one of these mutations, we're expressing Z in all of these, but now when we dial in one of the truncations, we suppress the morphological changes to the endoplasmic reticulum. And with the longer of those truncations, we actually have fewer endoplasmic reticulum changes than we saw with an MZ. So it seems to be better than being heterozygote. This is hinting at the fact that this is having a positive beneficial effect on these cells. This is all in cells, though. You only care probably about the liver. So if we go back to the liver samples, we can take sections next to the ones that Natalia used for her laser microdissection, and then stain with an antibody that recognizes polymers. So these black dots here, we know from Natalia's laser microdissection are portions that don't have a mutation. And you can see lots of nice brown staining, and they contain polymers. The green spot has one of these 5' mutations, one of the mutations which are probably just going to act as a null allele, going to remove the mRNA from the system. But when we look at spots that have these truncations, we initially got the impression that we were losing polymer accumulation from that cell. So what we did is we took many, many samples, we scored them blindly, and then when we broke the code, we saw if the level of polymers correlated in any way to the mutations. And what we see is that if any part of the liver that has acquired one of these C-terminal mutations, we have cleared polymers. So this is a beneficial effect. Expressing one of these C-terminal truncations helps the cell clear polymers. So almost finished, don't worry. We now have a situation where M is secreted, Z polymerized, but Z plus a truncation appears to be better than MZ. It appears to clear the polymers from the cell. We don't actually know precisely whether this is only a loss of toxicity or a dominant gain of benefit, but we have reason to believe that it is beneficial to the cell, and that might be useful for therapy in the future. So just to remind you, we have this truncation. It's lost the C-terminus, so it can't polymerize, or rather, it can't continue to grow a polymer because it lacks the C-terminus. But what it can do is it could accept the C-terminus of another molecule. What we've therefore done is the cell has evolved a version of antitrypsin that can cap a growing polymer and stop the polymer growing. Oops. I don't think I've gone over. If that's true, that means these C-terminal truncations should be able to interact with polymers and allow us to purify polymers from the cell. Again, unpublished data. If we take halo-tagged MZ or one of the Z truncations, express it in a cell along with Z, and then pull on the halo tag, so this is an affinity purification, do any of these proteins interact with the Z polymers within the cell? So we know that even M can incorporate into polymers. It just can't incorporate very quickly. So if we pull on M protein, it brings down a little bit of Z from the cell. If we pull on Z, not surprising, the Z halo tag, we pull down the Z polymers from in the cell. But what's really fun to us is that if we pull on these C-terminal truncations, we also pull Z polymers out of the cell. And if we look at the longer of those polymers, this is the one that has the reactive center loop forming the hyperstable conformation of alpha-1 antitrypsin, that brings down even more polymers. It's even more efficient at interacting with Z than Z is. This raises the possibility that we are generating this hyperstable trap to catch growing Z polymers, and that is somehow allowing the cell to dispose of them. Our current working model, and we're in the middle of these experiments at the moment, our current working model is that by making shorter polymers, we're generating a species which your hepatocytes are easier to clear. If that's true, then we've got two ways we might be able to use this therapeutically. One was we could introduce them. You could imagine a crazy model where you have an mRNA that you introduce into the hepatocyte to generate this material. We know that most of your mRNA vaccines end up in the liver. Why can't we use it as a way of delivering a truncated protein? Another is that it's possibly more acceptable would be to do mRNA editing. At the moment, drug companies are developing mRNA editing to either correct the Z mutation. That's a big ask. What we would suggest is all they need to do is cleave off the C-terminus, and then you'll generate a protein that can't polymerize and will allow the hepatocyte to clear the polymers it already has. So in the last half an hour, I've tried to convince you that in your patients, we are seeing evolution. In every one of the patients that get advanced liver disease, they are evolving mutations of in alpha-1 antitrypsin, which delete the C-terminus. And this means that rather than doing a genetic screen in animals or cells in the lab, we're actually doing it in patients. It's also suggested a completely novel therapeutic approach that we could use in order to clear these polymers. So thank you very much for listening. I've mentioned the 3 people who did the vast majority of the work. Obviously, I have to thank the funders who paid for this very expensive project, so thank you to them. We work within, the group is a nice collaborative group, and we're very happy to welcome any of you who want to visit at some point. So thank you for listening.
Thank you very much for your very good talk. I have 2 questions. The first thing is that, as far as I clearly understood, it seems that this additional somatic mutation could be somehow protective regarding the liver disease.
Yep.
But ironically, what gives you the tips is to study cirrhotic patients who were liver transplanted for alpha-1
Yep.
antitrypsin deficiency.
Yes.
So what could be the observation in alpha-1 patient without a severe liver phenotype?
This is a great question. Now, these patients, they have developed liver disease later in life. You could argue perhaps these somatic mutations mean that they come to your clinic in their 50s rather than in their 20s. That's one possibility. Gladly, we don't have many livers from patients early in the disease. So you don't need to refer for transplantation a 30-year-old person. So and unfortunately, not many of them give up their livers after car accidents. There aren't that many frozen biopsies. We need frozen biopsies to do this work. Now, I know we've talked today about the ability of doing sequencing on fixed samples. It's not very good for the sort of technologies that we need in order to detect these mutations. So I would say to anybody who has lots of frozen liver biopsies from patients who have earlier stage liver disease, I would love those, because we could sequence and we could find out how early these events happen.
Thank you. And maybe if I may have a second question, because you showed that this mutation decreased the aggregation and somehow could protect the liver, but at the same time, the secreted alpha-1 antitrypsin is decreased. So what could be the potential adverse event on the lung counterpart of this additional somatic mutation?
No, you're absolutely right. So remember, the evolution here is acting on the hepatocyte. The hepatocyte sadly doesn't care what the lung's doing. So the hepatocyte wants to live, and in a quite selfish way. Having said that, the best therapies that are currently, there's a lovely, Pavel Strand had a wonderful paper, is it New England Journal 2 years ago, 1 year ago, for RNA to knock it down. So we can keep the respiratory patients alive, okay? In most countries, not the UK, we can give alpha-1 antitrypsin as infusion, so you can do augmentation therapy. But what we don't have at the moment is any specific treatments for the liver. So a promising treatment would be RNAi, and that's been tried and reduces the polymer accumulation. Would be another way to remove the polymers. It's a really exciting time to be in alpha-1 antitrypsin research, because many drugs are in development. There are small molecule inhibitors for polymerization. There is RNAi, there is RNA editing, and there are even CRISPR. We don't know which one's going to work, but I've never seen this field with so many potential treatments. I think within 10 years' time, it will be really disappointing if you don't have specific treatments for this condition. Not for the lung, though.
Other question? Okay. I have one. We already talked together, but do you think that this form can be used as a biomarker to a good issue into the disease?
So, yeah, it would be lovely if we could detect it, because then I could answer. Sorry, I didn't get your name. What was it?
Emmanuel.
Oh, I didn't. If I could have answered this by saying, we can see this biomarker appearing in the blood. At the moment, we have no way of measuring it. So what we're looking at is and as we're talking with you, it would be a great idea to develop antibodies to the truncated form. We're also looking at cell-free DNA, because as liver diseases progress, more of the circulating DNA is actually from the liver. So we could look to see whether we can detect it there. So I think and also we're in discussion with Matthias Mann, to see whether we can do proteomics on the liver, on the circulation, to see if any of this protein goes out. So these would be biomarkers which would allow us to answer your question about when this happens. We would then be in a position to say, is a patient who develops these truncations early likely to live without developing liver disease as opposed to somebody who develops them late.